Hi guys, so moving on to our last learning unit here before our second exam, we're going to move on and talk about lipids. And this represents kind of the last of the biomolecules that we're going to study. So again, we studied nucleic acids first, then we spent several learning units talking about proteins. We just finished carbohydrates, and now we have lipids. So these kind of represent a unique class of biomolecules because they tend to be very water insoluble, so nonpolar substances. Even though we'll kind of talk about this a lot in terms of a linkage, we don't think of these as polymeric materials. But we are going to spend a little bit of time thinking about the same four categories that we talked about for all four of our other biomolecules. We'll talk about their information capacity, their ability to serve as an energy molecule, their ability to serve kind of in a catalytic role, and then kind of structural aspects. And so after we kind of have dealt with that, we move into kind of looking at some structural components. And so we'll talk about this and this and this. And once we kind of go through this material, it should make a little bit more sense. But again, this is going to be representing a fatty acid, which is the simple sort of building block of dietary fats and the way that we store fats in our body. And so we'll learn about the structures of fatty acids, and importantly, we'll learn about a lot of the different components of fatty acids, a lot of the different kinds of fatty acids. So saturated fatty acids, unsaturated fatty acids. We'll talk about omega fats. We'll talk about trans fats. So we'll spend actually a good bit of time at the very beginning part of this learning unit talking in great detail about the structure of many of these different fatty acids. Then we'll move on and we'll kind of think about, as we put these fatty acids together in a triglyceride, so again, 3 fatty acids hooked together on a glycerol backbone. We'll talk about the 2 potential sources of those: dietary fats, so things that you consume in your diet. That's generally about 95% of the fats that we consume are those kind of fats. But then we'll also talk about your body's synthesis of triglycerides and how that really is a proxy for excess levels of carbohydrate, particularly glucose, in your bloodstream. So we'll spend a good bit of time talking about that. We will talk about things like ketone bodies. So we'll talk about the way that your body can actually utilize fat reserves and make them into water-soluble molecules that we can circulate for fuel. So even though we don't necessarily think about this in terms of a linkage for polymerization, we do think about a linkage in that we have an ester linkage that hooks these 3 fatty acids onto our glycerol backbone. And so one of the things that we'll talk about is the condensation chemistry that allows us to make that linkage, and then the hydrolysis chemistry that breaks that linkage. So we'll have 2 reaction mechanisms that we'll examine for that. And again, this breaking part is really important because that's how we make soap, when we make soap kind of the old-fashioned way. And that's what we need to think about in order for you to consume dietary fats that come out of your digestive tract. So we'll move on after that, and we'll spend a good bit of time talking about cholesterol and sort of why cholesterol has sort of been vilified when it really is a very important biological molecule in your body, has important repair functions. Many of your hormones are produced from cholesterol as a precursor. So the importance of kind of having cholesterol and not vilifying it as a bad molecule. We'll wrap up sort of talking about some fat-soluble vitamins like vitamin D and vitamin K, and again, their role in this whole process of inflammation and cardiovascular disease. And then we'll also talk a little bit about some downstream uses of fatty acids. So the synthesis of prostaglandins for the regulation of pain, fever, and inflammation. So we'll kind of talk about those different things. And then at the very end, and in a way to lead into our next learning unit, which will be part of our next exam, we'll talk about our lipid bilayers. And so whether you have fatty acids that kind of aggregate to make micelles, or you have phospholipids that make lipid bilayers, the shape and the structure of these molecules kind of dictates their function. And so we'll talk a little bit about the hydrophobic effect again, and then how we can kind of have a structural component to these molecules.